Hello everyone, I am Doel Bose. This is the third lecture in the 4-part series of Demystifying Genetics. This is specifically meant for high school students, undergraduates, medical graduates, or even doctors who wish to brush up their genetics. In the first part, we covered some basic definitions, the central dogma, transcription, translation, and so on. In the second part, we covered cytogenetics, which was specifically meant for structure of chromosome, chromosomal errors, classification of chromosomes, etc. Today, in this third section, we are going to cover genes and genetic mutations. Genetics is just like mathematics. If you miss out on the basics, you will find it extremely difficult to understand the advanced concepts. So if you have not already gone through the first part, I would request you to first go through the basic definitions like nucleotide, DNA, and especially the transcription and translation part. So let us start today's session. Zooming in to the structure of a gene, we already saw that genes are a sequence of DNA. This is a simplified structure of a gene which shows two different types of segments called exons and introns. Introns are non-coding region of a gene, meaning this region is not transcribed. Exons are coding regions of the gene, that is the region which is transcribed. Easy way to remember is extrovert and introvert. Extroverts allow interaction, so exons allow transcription. What you see on the screen is a pair of homologous chromosomes. Location of a gene on a chromosome is called a locus. An allele is a variant or a version of a gene. Each cell has two alleles for a single gene, one inherited from each parent. So what do you mean by versions of a gene? If you remember the codon chart, the chart that we saw in our first lecture, I would request at this point that if you have not already gone through that, please go through it, because genetic mutations are difficult to understand unless you are well versed with the codon charts. On the codon chart, you saw that one amino acid can be coded in more than one way. So you can have more than one ways of coding a polypeptide chain. That is more than one version of a gene. Homozygous refers to an individual if they inherit similar versions of a gene at a specific locus of the gene, while heterozygous refers to an individual if they inherit different versions of the allele at a specific locus. Now coming to genetic mutations. Changes in the structure of the gene. By structure we mean the sequence of nucleotides and the number of nucleotides without changing the position of the gene is called as a gene mutation. Mutations can be classified on the basis of the effect of the mutation. So you could either end up having a loss of function, or it could be a neutral mutation, or it could be a gain of function. By loss of function, we mean it results in an inactive or a less active protein which is being coded. By neutral mutation, we mean that there is no net change in the protein which is being coded. And by gain of function mutation, we mean that it leads to a more active or a protein which has acquired a different function. Now classifying genetic mutation based on the region of the mutation, like we saw, changes in the gene sequence on nucleotides, typically which is less than 100 kb, is called a gene mutation. Now gene mutation could take place in either the protein encoding sequence or the transcription regulating sequence. If it impacts the protein encoding sequence, it impacts the protein synthesis. While if the mutation occurs in the transcription regulating sequence, for example in the promoter, enhancer, silencer, etc., this will impact the gene interaction and the gene regulation. And as a result of the disruption of the interaction between these molecules, abnormalities will occur in the cell. A mutation is a process of alteration in the genome sequence by mistake during DNA replication, or as a result of environmental stress including radiation, toxic chemicals, and smoke. DNA exposed to mutagenic chemical, where mutagenic meaning things that cause mutation like X-ray or ultraviolet radiation. Now coming to the classification of genetic mutation. Classification of genetic mutation is kind of difficult in a classification tree because of the overlapping elements. However, just for the sake of simplification of this lecture, we would stick to this broad classification of the genetic mutation in either a point mutation or a repeat expansion. Point mutations can be of two types, either a base substitution or an indel or insertion or deletion mutation. Base substitution is subcategorized into silent, missense and nonsense mutation. Coming to the base substitutions. Let us see what each of these mean. Consider a codon GAA that eventually translates to a glutamic acid. So what is the impact of a point mutation, meaning the mutation on the third position of the codon that changes it from GAA to GAG? So getting back to the codon chart. Since both GAA and GAG code for the same amino acid, that is glutamic acid, we see that there is no impact or no change in the effective end protein, and hence we call this a silent mutation. This is in fact a case of code redundancy where two different codes result in the same amino acid chain. And since there is no change in the amino acid chain, we call it a silent mutation. The next point mutation is a nonsense mutation. If the mutation resulted in changing the first position of the codon from a G to a U, causing GAA to change to a UAA, getting back to the codon chart, you end up coding for a stop codon. Hence, the transcription of the amino acid chain stops abruptly. The resulting amino acid chain remains shorter, therefore it is called a nonsense mutation. The third possibility is changing a GAA to a GAC in the codon. Let us see the impact of the change. As you can see, GAA codes for glutamic acid, whereas GAC codes for aspartic acid. This would completely change the amino acid that is translated from glutamic acid to aspartic acid, and hence this is termed as missense mutation. This leads to a change in the final protein. It is always more impactful to learn something through examples. Let us see some examples of genetic disorders caused due to point mutation. The first genetic disorder that we will be talking about is a Marfan syndrome. This is a nonsense mutation. Here, the point mutation results in a stop codon instead of coding for tyrosine amino acid, hence a nonsense mutation. This results in abrupt stop in the protein synthesis, resulting in a shorter than normal protein being produced, and this impacts the skeletal and cardiovascular system. The next example of point mutation is sickle cell anemia, which is kind of a missense mutation where the impacted gene is the beta globin. One change in the codon of the DNA strand from CTC to CAC results in a different amino acid to be coded. That is, instead of glutamic acid being translated, valine is formed. This results in sickle-shaped hemoglobin molecule. The next kind of point mutation is an indel mutation or insertion or deletion. Insertion or deletion of one or two nucleotide results in frame shift mutation. Frame shift mutations are insertions or deletions in the genome that are not in the multiples of 3 nucleotide. They are subset of insertion deletion or indel mutations that are specifically found in the coding sequence of a polypeptide. Here, the number of nucleotides that are added or removed from the coding sequence are not in the multiple of 3, and hence there is a change in the reading frame. They can arise from extremely simple mutations such as addition or removal of a single nucleotide. To understand a frame shift mutation, we should first see or understand what is a reading frame. Reading frame is a group of 3 successive base pairs that represent a codon. So the original reading frame was CCU, GAC, UAC, CUA. Deletion of nucleotide U results in the shift of the reading frame, and now the sequence is CCG, ACU, ACC, and so on. This results in translation of an entirely new sequence of polypeptide chain. Beta thalassemia is a classic example of a frame shift mutation. Beta thalassemia is caused by reduced or absent synthesis of beta globin chain of the hemoglobin tetramer. Hemoglobin tetramer meaning 4 polymers, is formed by 2 alpha polymers and 2 beta polymers. The beta polymer is coded by HBB gene on chromosome 11, whereas alpha polymers are coded by a gene on chromosome 16. In a normal person, there is a balance between production of alpha and beta gene, resulting to a balanced structure of the hemoglobin molecule. Deficiency of either alpha or beta globin chain results in instability, and the RBC formed are not of proper shape, so they cannot carry sufficient oxygen, resulting in anemia. In a normal case, one inherits 2 versions of chromosome 11 with 2 versions of the beta globin gene, which encodes for enough number of beta globin polymers, resulting in a balanced amount of globin molecules. But if one of the chromosome has a missing gene, or if there are mutations in the gene of one chromosome, it results in either beta thalassemia trait or beta thalassemia major. There are more than 250 disease-causing mutations which have been identified so far on the HBB gene. Majority of them are single nucleotide substitution, deletion or insertions which lead to a frame shift, and hence this is called a classic example of a frame shift mutation. Another example of frame shift mutation is cystic fibrosis. This is actually a result of 2 frame shift mutations in the CFTR gene on chromosome 7. This results in shift in the reading frame to introduce a UAA, which is a stop codon or termination codon. This results in severely truncated and non-functional protein. The CFTR gene regulates proper flow of ions, that is chloride and sodium, across the cell membrane of lungs and other organs. So this brings us to a concept or a term that you must have heard plenty of times, SNPs, single nucleotide polymorphism. By the name, we can say that single nucleotide polymorphism is change in one nucleotide, right? So how is it different from a point mutation of a single nucleotide, or is it the same? So by definition, single nucleotide polymorphism as shown on the screen, when you have change in just one nucleotide, for example, here we have shown a change from an A to a G in 2 different people, then this is a SNP. An alteration of a single base results in sequence modification in the amino acid, or it could be a neutral, or it could be a stop codon, like we saw in our example, right? And this could result in either no change at all or a malfunction of a corresponding protein. So when do you call it a SNP and when do you call it a point mutation? If the single base change occurs in less than 1% of the population, you call it a point mutation. But if the single base change occurs in a population at a frequency of 1% or higher, you call it a SNP or single nucleotide polymorphism. The next type of genetic mutation is repeat expansion. Many parts of human genome have repeated sequences. It is common to have parts of genome with few repeated trinucleotides like CAG or CGG, as shown here in the screen. Trinucleotide repeat expansion occurs when the number of triplet repeats present in the mutated gene is greater than the threshold number found in a normal gene. They are also called microsatellite expansions. In a trinucleotide repeat expansion mutation, the number of trinucleotide repeats increase in copy number until they cross a threshold value, above which the gene becomes unstable. Repeat expansion at time causes formation of hairpin structure, which makes the mRNA molecule unstable. The expansions could be present in either the coding region of the gene or could be present in the non-coding region of the gene. Depending on its location, the unstable trinucleotide repeat may cause defects in a protein encoded by the gene, or change the regulation of the gene expression, or produce a toxic RNA, or lead to a chromosomal instability. Trinucleotide repeat expansions, or TNR, mostly cause neurodegenerative diseases. Examples of repeat expansions to the coding region are Huntington's disease and Kennedy disease. Both of these are caused by CAG repeats. Example of repeat expansion to the non-encoding region are Fragile X and myotonic dystrophy. Over 50 genetic disorders have been identified so far that are a result of TNR or trinucleotide repeats. This almost brings us to the end of our presentation. Let us quickly look into the classification of genetic disorders by the number of genes affected. We can classify the genetic disorders as either single gene disorder or polygenic, meaning when multiple genes are involved in manifestation of the disease. Single gene disorders are also known as Mendelian's disorder. They are the most well-studied genetic disorders. So far, we have identified close to 7,000 Mendelian disorders impacting roughly 400 million people worldwide. Some facts about genes and genetic disorders. These are fascinating for sure. The genome of any two people differ by less than only 1%. H4C16 is the smallest gene that encodes the protein H4, histone H4. This gene is intron-less and is 406 base pair long. The largest gene, DMD or dystrophin gene, is 2400 kbs. There are 17,289 variants of the DMD gene, and it takes about 16 hours to make dystrophin protein in our body. And lastly, here are my references. I cannot thank search engines enough, and they have always helped me in my research and presentation. Thank you everyone for your time, and thank you so much for your patient hearing.